Bacterial vitamin B12 metabolism under different temperature regimes. Elise Schofield, Margaret Mars Brisbane, Harriet Alexander, Max Saito. Background. Vitamin B12 is a key driver of marine microbial community dynamics. Its availability limits the ability of different organisms to grow and develop. One important B12-driven process involves the enzyme methionine synthase. Methionine synthases catalyze the biosynthesis of an amino acid called methionine, which in turn acts as a building block for other amino acids and proteins. There are two different versions of this enzyme that organisms encode for: MET-H, which is vitamin B12 dependent, and MET-E, which is independent of vitamin B12. Vitamin B12 is produced by about 1/3 of prokaryotes, although many prokaryotes that cannot produce B12 still require it. This means that the majority of microbes rely on an exogenous source of B12, and that B12 is a limiting resource in the environment. Many phytoplankton require vitamin B12 to grow, but no eukaryotic phytoplankton can produce the vitamin themselves. So these phytoplankton must rely on B12 synthesizing bacteria. Objective. This study looks at B12 metabolism in 2 bacterial species under different temperature conditions in order to investigate how increasing ocean temperatures affect B12 metabolism, and as a result, how temperature change could impact microbial communities. 2 species with contrasting abilities to synthesize vitamin B12 and contrasting B12 requirements are included to determine if warming temperatures will differentially impact bacteria with distinct B vitamin metabolisms. Bacteria. Altermonas macleodii does not synthesize B12. Instead, it relies on exogenous sources of B12. Encodes for both MET-H and MET-E methionine synthases. Particle and phycosphere associated, can be found in temperate tropical oceans. Wucheria pomeroy synthesizes B12. Provides an exogenous source of B12. Encodes for MET-H methionine synthase. Phytoplankton symbiont. Can be found in tropical oceans. Figure 1 illustrates latitudinal and temperature ranges of A. macleodii and R. pomeroy based on Tara Ocean's metagenomic sequencing reading map. The left graphic shows this information for Altermonas. The right graphic shows the same information for Wucheria. The x-axis represents the latitude group on a scale from -60, increasing by 10, to +50 degrees. The y-axis represents the log-scaled sum RPKM. For the Altermonas graph, the scale ranges from e to the -2 through e to the +2. For the Wucheria plot, however, the scale ranges from e to the -4 to e to the +0. The mean temperature in degrees Celsius is represented by a color gradient, with 0 degrees Celsius represented by blue, and values over 20 degrees Celsius represented by red. Methods. Altermonas macleodii and Wucheria pomeroy were inoculated into 3 different media types: unaltered minimal media, media without vitamin B12, and media without any B vitamins, including thymine, biotin, and B12. Each combination was then tested across a temperature gradient ranging from 12 degrees Celsius through 42 degrees Celsius. After incubating in the thermoblock, cell concentrations were measured at timed intervals using a flow cytometer. Mass spectrometry will be used to measure how much B12 is used, stored, and produced by Rogéria pomeroy at different temperatures. Results. Trial 1. Control versus no B12. Figure 2 illustrates the growth curves for A. macleodii and R. pomeroy grown at different temperatures in unaltered minimal media, full, and media without B12, minus B12. Points are mean cell counts from 3 biological replicates, and error bars represent one standard deviation of the mean. B12 availability limited total growth at the highest temperatures, but there was not enough data to calculate growth rates. Maximum temperatures and measurement time intervals were adjusted for trial 2, with the maximum temperature for R. pomeroy decreased and the first measurement taken closer to the time of inoculation. The left side of this graphic shows the data for AMAC, while the right side of this graphic shows the data for R. pom. The x-axis represents hours from inoculation, with a scale increasing by 10 from 0 to 50. The y-axis represents cells per microliter, with the scale increasing by 20,000 from 0 to 60,000. Temperature gradients are represented by colors, with the cold gradient represented by blue, the middle gradient represented by yellow, and the hot gradient represented by red. The shape of the data points is also indicative of the media used. The circle data points represent the full minimal media, and the triangular points represent no B12. Trial 2. Control versus no B vitamins. Figure 3 illustrates growth curves for A. macleodii and R. pomeroy grown at different temperatures in unaltered minimal media, full, and media without vitamin B12, thiamine, or biotin, minus B vitamins. Impact on total growth was similar to what was observed in trial 1. Availability of B vitamins limited total growth of A. macleodii at high temperatures. The mu max still could not be accurately calculated due to having too few data points during exponential growth phase. The x-axis represents hours from inoculation, increasing by 10 from 0 to 30. The y-axis represents cells per microliter, increasing by 20,000 from 0 to 60,000. The left side of this graphic shows the data for AMAC. The right side of this graphic shows the data for R. pom. The temperature gradients are also represented by color, with the cold gradient represented by blue, the middle gradient represented by yellow, and the hot gradient represented by red. The shape of the data points is also indicative of the type of media the bacteria was inoculated into, with circle representing the full minimal media, and the triangles representing the no B vitamin media. Trial 3. High-resolution control versus no B12. Figure 4 shows the growth curves and maximal growth rates for A. macleodii and R. pomeroy grown at different temperatures in unaltered minimal media and media without B12. Impact on total growth was similar to what was observed in trial 1 and 2. Availability of B12 limited total growth of A. macleodii at high temperatures. A. macleodii mu max was depressed in media not supplemented with B12. The black line with a triangle represents the time when the R palm was harvested for mass spec measurements of the B12. The rightmost graphic shows the maximal growth rates for A. macleodi and R. pomeroy. The left side represents A. mac, while the right side represents R. pom. The x-axis shows temperature with the cold gradient, the middle gradient, and the hot gradient. Media type is indicated by color, with the green representing full media and the red representing no B12 media. The y-axis shows the maximum growth rate, or mu max. Conclusions. Availability of exogenous B12 and of B vitamins, B12, biotin, and thiamine, decrease the total growth of A. macleodi at high temperatures in all 3 trials. Availability of B12 reduced the growth rate of A. macleodi, but not R. pomeroy, suggesting that when B12 is limiting, the ability to synthesize B12 supports higher growth rates than having B12 independent methionine synthase. Ongoing work will determine whether temperature influences B12 production by R. pomeroy. Future questions. Of the vitamin B12 that is produced by R. pomeroy, how much is used and how much is made available to symbiotic phytoplankton? How do B12 synthesizing bacteria, such as R. pomeroy, transfer B12 to symbiotic phytoplankton? How will changing temperatures influence the bioavailability of vitamin B12, and how would that impact phytoplankton dynamics and upstream food webs? What are the ecological outcomes if overall abundance of certain bacterial taxa change? For instance, how will ecosystem function change if B12 synthesizing bacteria outcompete other bacteria? Acknowledgements. Dr. Julie Huber, Dr. Greta H. Serres, Ariana Krinos, Dr. Matthew McIlvin, and Dr. Erin McParland. Center for Chemical Currencies of a Microbial Planet. Woods Hole Oceanographic Institution. Massasoit Community College. The National Science Foundation. Thank you for listening to this poster session. Feel free to reach out to the author on Twitter @EliseHunter or through email at alschofie2@gmail.com.